Hi guys, welcome back to my channel, and in this video guys we will be studying about restriction fragment length polymorphism. I'm just showing you that board in this figure so that I can clearly explain you by standing in this position and how is this process basically taking place. So restriction fragment length polymorphism guys, it is a type of a genetic marker technique. In a genetic marker technique, what does RFLP stands for? First of all, it is standing for the restriction fragment length polymorphism. In this, the first step is that we are isolating the DNA. But this stepwise method which I have drawn on the board, which this we will be taking on a look a little later. First of all, let us understand what is RFLP. It is a genetic marker technique in which the organisms are differentiated. How can the organisms be differentiated? By the analysis of the patterns derived from the cleavage of their DNA. What happens here in this process, we are taking restriction endonuclease enzyme, and this restriction endonuclease enzyme helps in creation of different patterns, and different patterns are created when the cleavage is being taking place at different positions. So it is a genetic marker technique in which the different organisms, we can differentiate the different organisms on the basis of their analysis patterns created by the cleavage of their DNA. जिस भी एंगल से वो DNA कट करता है, उसकी क्लीवेज जिस पॉइंट से होती है, वहाँ से लेके वहाँ तक एक डिफरेंट पैटर्न फॉर्म होता है, जिसकी वजह से हम उसको डिफ्रेंशिएट कर पाते हैं. This technique is very important as it takes an advantage of the difference in the DNA sequence. Difference in the DNA sequence कैसे आता है? With the help of an enzyme called as restriction endonuclease enzyme. If the two organisms, अगर दो organisms हम लेते हैं, if they are differing in the distance between the site of their cleavage of a particular restriction endonuclease, the length of the DNA fragment produced upon ligation will always be different. राइट और अगर सिमिलर पैटर्न्स आते हैं तो हम उसको ईजिली यूज़ कर सकते हैं ताकि हम ईजिली दो डिफरेंट स्ट्रेन्स ऑफ स्पीशीज़ को सेपरेट कर पाएँ एक दूसरे से. तो रिस्ट्रिक्शन एंडोन्यूक्लियस इसमें क्या करता है? रिस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम क्या करता है? डबल स्ट्रैंडेड डीएनए को डाइजेस्ट करता है एक स्पेसिफिक साइट पे, जनरेट करता है कलेक्शन ऑफ डीएनए फ्रैगमेंट्स जिनकी पॉसिबली जो लेंथ होती है वो सेम होती है. जैसे कि हमने फर्स्ट टू फिगर्स में हम देख रहे हैं, इन द होल फ्लो चार्ट इन द फर्स्ट टू फिगर्स यू कैन इजीली सी दैट वी हैव आइसोलेटेड द डीएनए फ्रैगमेंट्स, and now these DNA fragments are exposed to restriction endonuclease, which in turn are resulting into formation of fragments. These tiny fragments which are present, ये fragments जो होती हैं ये nearly equal in number होती हैं. अब कैसे वो fragments को ligate करता है, digest करता है? If the bases ATGC of the DNA are evenly distributed with the same frequency, the enzyme having the recognition of site of 6 base pairs, तो वो DNA को हर 4096 base पे काटेगा. In this method, what is happening now? Gel electrophoresis is being taking place. In the gel electrophoresis, they are separated on the basis of their size, on the basis of their pattern which is being created by the restriction endonuclease, and after that we are doing the alkali treatment also so that the DNA is denaturated. Then the next step is the sudden blotting. After the sudden blotting, we are also applying the probe, and then washing of the probe is done, which is not reacting to it. After that, the process is done, and this is known as the standard RFLP, that is restriction fragment length polymorphism. So I hope you guys have understood this video very clearly. In this, it is a simple process, a very normal process in which restriction fragment length polymorphism has been shown. Right, I hope you guys like the video guys. Stay tuned for the next video, and thanks for